Good morning everyone. Welcome to today's tutorial for biology. So today we'll be looking at tutorial number 6, which is on nucleic acids. So before we start, remember to change your settings so that you have the best quality. Go to your quality settings and change them so that you have a good resolution. Okay, so I'll just give you a few moments to do that. Okay, so now we can start. So tutorial sheet number 6 is on nucleic acids, as I said earlier. So we'll start with the first question. Question 1 reads, state the 2 different groups of sugars found in nucleic acids. I'll read the question again. State the 2 different groups of sugars found in a nucleic acid structure. Okay, so in nucleic acids you find 2 groups of sugars. They are both pentose sugars, and these are, so the first one you find is ribose, which is found in ribonucleic acids, RNA. And the second one you find, the second sugar you find in nucleic acids is deoxyribose. Which is found in DNA. So both of these sugars are pentose sugars, meaning they have 5 carbon atoms in their structures. And you find ribose in RNA, which is ribonucleic acid, and you find deoxyribose in DNA, which is deoxyribonucleic acid. Okay, so that answers question 1. Then we go to question 2. Okay, so let me read question 2. Question 2 reads, what are purines and pyrimidines? What are purines and pyrimidines? So we're being asked 2 questions here. Purines and pyrimidines. Okay, so we'll start by talking about purines. So now, what are purines? So purines are heterocyclic aromatic compounds that consist of 2 rings. So I repeat, purines are heterocyclic aromatic compounds that have 2 rings. So examples of purines are adenine, and guanine. Okay, so let me explain a bit. So what does heterocyclic and aromatic mean? So heterocyclic An heterocyclic compound is simply a compound that is made out of, that has ring structures. So that has a ring structure. So we are saying purines are heterocyclic. It means they are ring structures. They are compounds that have a ring structure. Then aromatic is a planar, which means a plain molecule which is unsaturated. A plain molecule which is unsaturated. So in other words, when we say purines are heterocyclic aromatic compounds, we are saying they are cyclic compounds which have unsaturated atoms in them. Then for something to be referred as a aromatic, it has to be made out of at least, this structure has to have, the backbone of this structure has to have at least 2 different elements. So for purines, the 2 different elements that make up the backbone of each structure are carbon and nitrogen. Carbon and nitrogen. So examples are adenine and guanine. They each have 2 rings. Their structures both have 2 rings, and these 2 rings are unsaturated. So we know that an unsaturated compound, it simply means there are double bonds in that compound. That's what we mean by unsaturated. Okay, so those are purines. Now, what are pyrimidines? Pyrimidines So pyrimidines are also heterocyclic. They are also heterocyclic aromatic. Aromatic compounds. Now, for them, they only have 1 ring structure. One ring structure. So what are some of the examples of pyrimidines? So examples are 1, thymine, uracil, cytosine, and what else? Okay, yeah. So those are some of the examples. Many others exist, but I've highlighted this because they are of relevance when it comes to nucleic acids. So these are some of the pyrimidines that exist, and these are the ones that are important for nucleic acid. Okay, so I believe we have answered question 2. Okay, so another thing you have to take note. So both purines and pyrimidines are referred to as nitrogenous bases. Nitrogenous bases because they have basic properties. They have basic properties. So remember that both of them are referred to as nitrogenous bases. Nitrogenous bases. So this is because they have basic properties. Okay, so we can move on to the next question. So question 3. Question 3 reads, state and draw the 5 different bases that are found in nucleic acids. State and draw the 5 different bases that are found in nucleic acids. Okay, so What are the 5 nitrogenous bases that are found in nucleic acids? Okay, so we'll start with the pyrimidines. So there are 3 pyrimidines that are found in nucleic acids and 2 purines that are found in nucleic acids. So we are starting with the pyrimidines. So the first one we are going to look at is cytosine. Others call it cytosine, others cytosine. They can't pause.
Just continue.
Okay. Okay, sorry for the disturbance. We're having some technical issues. Okay, just a minute.
It's the way like you are here. There is a sound that is coming from both waters and quantities. It's like
So apologies for that. We were having some technical difficulties. I hear there was a problem with sound. I hope it's been sorted now. Okay. So yes, I was talking about cytosine. So yeah, the question is asking us to mention the five bases that are found in nucleic acids, and we are also supposed to draw their structure. So the first one we'll look at is cytosine. Okay. So if you remember in the definition, we said they are aromatic in nature, which means they have double bonds and their backbone, the ring structure, is made out of of 2 or more, at least 2 different types of elements. And we said for purines and pyrimidines, the elements are carbon and nitrogen. So the backbones are entirely made of carbon and, sorry, let me just draw it here. Okay. Okay, so this is the structure of cytosine. So we see the backbone of this, the ring structure, is made out of 2 different types of atoms, which are carbon atoms and nitrogen atoms. So when we are talking about nitrogenous bases, we number the atoms in the ring structure starting from the one down here. So this is the first atom. This will be considered as the second. This one the third, the fourth, the fifth, and the 6th. So there are there are 6 atoms in a pyrimidine structure. So the ring structure is made out of 6 carbons, and out of these 6 we have 4 of them are carbon atoms, and the other 2 carbons are nitrogen. So this is the structure of cytosine. Okay, so now we'll now look at the structure of thymine. Thymine, so it's also a okay. So this is thymine. That's the structure of thymine. So here this is our first carbon, second, third, fourth, fifth, and our sixth. Okay, so that's thymine. Then we now go to the last pyrimidine that we have, which is uracil. Okay. So for uracil, it's very identical to thymine, and the only difference is that on carbon number 5, we don't have a methyl group attached. Instead, we just have a hydrogen atom, a hydrogen atom attached there. Okay, so this is your uracil. So these are the 3 pyrimidine structures that exist. Yes, so now we will now go to the purines. We'll now go to the purines. So we've seen that there are 3 different pyrimidines. There are 3 different pyrimidines that are found in nucleic acids, but for purines we only have 2. So we have adenine, so the structure of adenine, Okay, so that's adenine. Then in purines, like in pyrimidines, we start numbering our atoms from this nitrogen here. So this will be atom number 1, 2, 3, 4, 5, 6, 7, 8, 9. So in a purine molecule, we have 9 atoms in the ring structure. So remember, by definition, the purines have 2 ring structures. So we have 1 and 2. So these are the 2 ring structures that are found in purines, in adenine specifically. So that's the structure of adenine. So now we will now go to the structure of guanine. Okay, so again we number the atoms. So in guanine, this is 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so these are the two different purines that exist. Okay, so these are the structures of guanine and adenine. Okay, so we are done with this question. We move on to the next question. So we'll move on to the next question. So I'll read the question. So we're now on question 4, which reads, illustrate the condensation process involved in the synthesis of a nucleotide. Okay, so a quick revision. We know when a nitrogenous base is joined to a ribose sugar, which is either ribose or deoxyribose, you form what's known as a nucleoside. Now, when a nucleoside is joined to a phosphoric acid or a phosphate group, you are going to form what's known as a nucleotide. Okay, so what we are being asked to illustrate is the formation of a nucleotide. So take note of that, not a nucleoside. So in other words, formation of a nucleotide is simply the joining of a phosphoric acid to a nucleoside. Okay, so let me write that down. So when you join a phosphate group, plus a nucleoside, what you are going to have is known as a nucleotide. Yes, so don't confuse these two terms, nucleoside and nucleotide. Don't confuse those two terms. Okay, so now we are going to illustrate how a phosphate group can be condensed to a nucleotide. to a nucleoside to form a nucleotide. Okay, so we're going to illustrate how this happens. So the phosphate group that is involved is the phosphoric acid, which is simply a phosphate Okay, so this is phosphoric acid. So we are going to join this to a nucleotide, a nucleoside, sorry. So we can choose any nucleoside. So for example, let's say we are working with adenine. So we know that in a nucleotide we have, in a nucleoside, sorry, we have a pentose sugar. Okay, so we have our pentose sugar there. Pentose sugar, which is attached to Okay, so we have our nucleoside. Our nucleoside here, which is a pentose sugar attached to an adenine. So this nucleoside is known as adenosine. So this is known as adenosine. So we have our phosphoric acid plus our nucleoside, which is adenosine. So where is the reaction going to happen? The reaction is going to happen between, so if you remember, in a pentose sugar, we start counting our carbon atoms from this one, which is the carbon atom which has the functional group. So this is 1, 2, 3, 4, 5, carbon number 5. So the reaction is going to happen between the hydroxyl group on carbon number 5 of the pentose sugar and the hydroxyl group of the phosphoric acid. So we'll have a wonderful reaction here, which will yield a water molecule. So the water molecule is going to be released from there, and we are going to form a phosphoester bond, phosphoester linkage. So we are going to illustrate that here now, what we are going to have. Okay, so when this water molecule comes off, so we form a phosphoester bond here. So what we are going to have is so we see this arrangement. A phosphoester bond plus our water molecule. Okay. So this is the process that is involved in the synthesis of a nucleotide. So now that a phosphate group has been added to our nucleoside, we now have what's known as a nucleotide. So this nucleotide is what's known as adenosine monophosphate. Adenosine monophosphate. So another phosphoric acid can be added to this molecule. If another one is added, it will be known as adenosine diphosphate, and if another one has been added, it will be known as adenosine. So if three of them have been added, it will be adenosine triphosphate, which is commonly known as ATP, which you are all familiar with. Yes. So, but in nucleic acids, only one phosphate group is added to form adenosine monophosphate. Okay, so this is how we illustrate our reaction, formation of a nucleotide. Okay, so we are done with that question. We can now go to question number 5. Okay, let me read question number 5. Question number 5 reads, explain the difference between a nucleoside and a nucleotide. Okay, so it's basically to explain what we are just talking about. What's the difference between a nucleoside and a nucleotide? Okay, what's the difference between a nucleoside and a nucleotide? So, yeah, we have been asked to say the difference between a nucleoside 1 and 2 nucleotide. So in a nucleoside, what we have is a pentose sugar, we have a nitrogenous base, okay, so if you have a pentose sugar plus a nitrogenous base, what you have is a nucleoside. Then a nucleotide, you have a pentose sugar you have your nitrogenous base, and you're also going to have a phosphate group. Okay, so this is what differentiates nucleosides from nucleotides. So in nucleotides we have phosphate groups which are not present in nucleosides. So that's the point which you have to remember. That's the major difference between nucleotides and nucleosides. Okay, so we move on to the next question, which is question 6. Okay, for question 6, it reads, what is the significance of nucleotides in a chromosome? What is the significance of nucleotides in a chromosome? So we know what a chromosome is. A chromosome is a strand of DNA. So a chromosome is a strand of DNA. So DNA is deoxyribonucleic acid. So we know that each So in this DNA, the building blocks of DNA are what are known as nucleotides. So each individual nucleotides can be added to another, and they are added to each other and form a long strand, which is known as DNA. So now we're being asked what the significance of these nucleotides are in DNA. So now, the first thing you have to know about nucleotides is that the sequence in which, so the first significance is the sequence of the nucleotides codes for a particular protein. Yes, so for example, if you have an adenine, then after the adenine, thymine, and guanine, so 3 nucleotides, they code for a particular protein. So again, another combination, you can have maybe CCA. So another, so each three nucleotides in a DNA strand code for one protein. So 3 nucleotides put together, they are referred to as a codon. So they are referred to as a codon because 3 nucleotides in a sequence will code for a particular protein. So yeah, so the first importance you have to remember is, the first significance is that the sequence of the nucleotide will code for a particular protein. Okay, so another major significance, so we know that the human body's the currency of energy in the human body is what? ATP. So ATP, which is adenosine triphosphate, is used to generate energy. For cellular activity. So ATP, adenosine triphosphate, is used to generate energy for different cellular processes in the cell. Okay, so those are the major significances of nucleotides in the body. Okay, so now we can move on to the next question. So if you look at the tutorial sheet, unfortunately there was no question 7. Question 7 was skipped. So we'll go to question 8. Question 8. Question 8 reads, illustrate how a DNA molecule is built up with the nucleotides and phosphates. Okay, so in question 5, we looked at how in question 4, we looked at how nucleotides are formed, but this time we will look at how nucleotides are joined together. Yes, so to illustrate this, we'll have 2 nucleotides. Okay, so let's say we have 2 nucleotides, and we know each nucleotide has a sugar, a pentose sugar, so joined to a nitrogenous base. Okay, so we use this to represent a base. Let's say maybe this is this is let's say adenine, Okay. We have our sugar. And we have our phosphate group there. Okay, so we have one nucleotide there. Then we have another nucleotide here. So again, this is our okay. This is our sugar. Okay, so due to space, I want to draw to scale. Okay, so let's say this time it's connected to thymine. This is thymine. We have our O there, our O. Okay. Okay, so we have two nucleotides. We've got two nucleotides there. So now, if you recall, in a pentose sugar, we start numbering our carbons from here. So this is 1, 2, 3, 4, 5. So now the reaction is going to happen between the phosphate group, which is connected to carbon number 5, and the hydroxyl group, which is connected to carbon number 3 in the other nucleotide. So now, because this phosphate group is connected to carbon number 5, this part of the molecule is known as the 5' end. 5'. And because this hydroxyl group is connected to carbon number 3, this part of the molecule is known as the 3' end. Okay, so you take note of that. So now, the reaction is going to happen. The hydroxyl group will react with the hydroxyl group on carbon number 3. So it's 1, 2, 3, 4, 5. So this hydroxyl group is the one that takes part in the formation of DNA. So we'll have this reaction. So a water molecule will be given off. Then what we are going to have is okay, so I'm not drawing this to scale because we don't have space. Okay, so we'll have our first nucleotide there, which will be joined here at the carbon number 3, which will be joined to the other nucleotide. Okay. Okay, so this is the arrangement that we are going to have, and this linkage is referred to as the phosphodiester linkage. Yes, so this phosphate group, because there are two ester bonds, that's why it's diester. So it's referred to as the phosphodiester linkage. So the two nucleotides will be joined together by a phosphodiester linkage. So that is how the nucleotides are joined together in the synthesis of a DNA molecule. Okay, so that's question 8. So we now go to question 9. So question 9 reads, discuss the structure of DNA and RNA and explain the difference in the structure and functions between DNA and RNA. Okay, so I read the question again. Discuss the structure of DNA and RNA and explain the difference in structure and function between DNA and RNA. Okay, so we begin by discussing the structure of DNA. Okay. So we know that the basic units of DNA are nucleotides. So a DNA molecule is made out of two strands of nucleotides. So it's referred to as a double-stranded molecule. So the DNA molecule is one double-stranded then it forms a helix. So to illustrate, so the structure of DNA will look something like that. Okay, I'm not an artist, but I'm trying to illustrate the helix. Okay, yeah, so we should remember that the structure of DNA is double-stranded, then it forms a helical in structure. So that means there are two strands of nucleotides in a DNA molecule. Then apart from that, in a DNA molecule, you only find 4 types of nitrogenous bases. So the 4 types of nitrogenous bases that are found in DNA are adenine, cytosine, thymine, and guanine. Yes, so these are the only nitrogenous bases that you will find in DNA. Okay, so now what's the structure? So, and DNA molecules are very long. They are very long such that in a human body, they are packaged, or not just in the human body, in organisms, they are packaged in what's known as chromosomes. Because if you just leave them stretched, it will cause confusion. So that's why they are tightly packed to form chromosomes, because they are very long molecules. So that's the structure of DNA. Then the basic structure, so we'll now talk about RNA. So what's the structure of RNA? So now, RNA, first thing you have to know is RNA is single-stranded. Single-stranded. Then these are very short molecules compared to DNA. DNA are very long, so RNA, they are short. Then the types of nucleotides that are found in RNA. So, and another thing I forgot to mention, in DNA, the pentose sugar found in DNA is deoxyribose. So in RNA, we find, first of all, the types of bases that are found here are adenine, guanine, cytosine, and uracil. Then the pentose sugar here is ribose. Ribose. Remember, in DNA, the pentose sugar is deoxyribose. Okay, so this is what makes RNA different from DNA. The pentose sugar is ribose. Then the nucleotides found here are adenine, guanine, cytosine, and uracil. Then these are very short strands, and they are single-stranded. They are not a double helix or double-stranded. Yeah. So that's the structure of RNA. So now, what are the functions of RNA and DNA? So as we all know, DNA carries the information. So DNA carries all the genetic information. So that's information that's required for an organism to develop, live, and also reproduce. So it carries the genetic information, which is information that dictates development and growth and reproduction. So each cell in the body contains the genetic material. So so that's the major function of DNA. It carries the genetic material which is necessary for growth, development, and reproduction. So this genetic material will issue out instruction. So the genetic material, for example, in eukaryotic cells, is in the nucleus. So it's going to issue out instructions that will, for example, protein synthesis. Protein synthesis is coded by DNA, but the DNA won't leave the nucleus for it to go out and make sure protein synthesis happens. So what happens? The instruction will be issued out in the form of RNA. So when a particular sequence of DNA has to be transcribed, has to be translated. First of all, it will have to be transcribed into RNA. Then this RNA now is going to be translated into a protein. So we have our DNA. Our DNA will be transcribed. Transcription will have RNA. Now this RNA is going to be translated into a protein. Okay. So those are the functions of DNA and RNA. So in other words, RNA is simply an intermediary between DNA and the protein. So the information to create a protein is in the DNA, but the DNA will not leave the nucleus. So what does it do? It is transcribed into RNA. Remember, RNA are short sequences, short strands. So it will just a short strand of the information from the DNA will be turned into an RNA molecule. Since it's small and short, it can easily move out of the nucleus and go to the ribosome, where it can be translated into the protein. So that's the function. The function of the RNA is an intermediary between DNA and the product which it's coding, the DNA codes for. Okay, so that answers that question. Then we now go to question number 10. So question number 10, it reads, what is meant by the phrases antiparallel strands and complementary bases? What is meant by the terms antiparallel strands and complementary bases? Okay, so on question number 10. So question number 10, we have been asked to describe two terms. One is anti-parallel strands, and the other one is complementary bases. Okay, so antiparallel strands. Okay, so we said DNA molecules are double-stranded. So we have, let's say this is our DNA molecule. It's a double helix, so we have our double strands. So now, in a DNA molecule, remember when we talked about the phosphate group being attached to carbon number 5 of a nucleotide, and carbon number 3 is there's an hydroxyl group on carbon number 3. So on carbon so the end, so let's say the last amino acid for this strand is ending with a phosphate group. So if this strand is ending with a phosphate group, this side of the strand will be referred to as the 5' end. This strand will be referred to as the 5' end. So which makes this other end, its other end. Okay, so for illustration purposes, let me draw them straight so that you're able to see. For illustration purposes, but we know that in actual sense that it's a double helix. So we have one strand there and the other strand there. So if this strand ends with a phosphate group, this end of the strand will be referred to as the 5' end, and this side a 3' end. So now if one strand is arranged in this manner, it will mean this strand, for this strand, this end will be the 5' end, the 3' end, and this end will be the 5' end. So because they are parallel to each other but inverted. So because they are parallel to each other but inverted, we refer to them as anti-parallel. So the strands in DNA are anti-parallel to each other because they are inverted. So the 5' end of one strand will be next to the 3' end of the other strand, and so on both ends. So that's why we call them anti-parallel. So what causes this? What causes this is because of the way the bases between, so the bases, the nitrogenous bases between these two strands form hydrogen bonds with each other. So adenine bases will only form hydrogen bonds with thymine. that's in DNA. Adenine will only form hydrogen bonds with thymine, and guanine will only form hydrogen bonds with cytosine. So this is what's known as base pairing. So hydrogen bonds will only form between adenine and thymine, and guanine and cytosine in DNA. So if it's a DNA molecule, during transcription, adenine can also be paired with, so adenine can be paired with uracil for RNA, but for DNA, adenine with thymine and guanine with cytosine. So this is our base pairing, complementary bases pair. Yeah, this is our complementary bases pair. Okay, so that answers question 10. Then we go to the final question, which says explain the importance of nucleic acids in living bodies. Explain the importance of nucleic acids in living bodies. Okay, so we know that nucleic acids are simply, so the two types of nucleic acids that we find in our living bodies are RNA and DNA. Okay. So for DNA, the importance cannot be overemphasized because we know it carries all the genetic information which is necessary for human growth and development and reproduction. So that's very vital. So it carries hereditary information. In fact, DNA codes for everything, everything that we have. So our phenotype is coded by our DNA. So our structure, our skin color, eye color, our height, weight, everything is determined by our genetic information. So the importance of DNA cannot be overemphasized. Then for RNA, without RNA, our DNA cannot be expressed. So DNA is only able to be expressed because that portion of DNA which is supposed to be expressed will be converted into, or will be transcribed into a RNA molecule, which is then going to be expressed as a protein later on. Okay, so on that note, we've concluded tutorial sheet number 6, and that's where we end our tutorial for today. Thank you very much.